I'm John Strum, and this is Real Talk MS. It's July 15th, and we have a lot to talk about. A couple of months ago, in episode 401, we were talking about positive results being reported out of a phase 2 clinical trial investigating the effect of vidofludimus calcium on people with progressive MS. That therapy is being developed by biotech company Immunic Therapeutics, and today, Jason Tardio, the president and chief operating officer at Immunic Therapeutics, is joining me to share some of the details about that clinical trial. And Jason's also going to explain why Immunic believes that in addition to slowing time to disability, vidofludimus calcium also provides neuroprotection for people with MS. And that's a potential game changer. But before we get to my conversation with Jason Tardio, there are a few other things that you should know about. In last week's episode of the podcast, I shared what I consider to be some very disturbing information from the Yale School of Public Health and the Leonard Davis Institute of Health Economics at the University of Pennsylvania about how the $1 trillion cut in Medicaid funding that was signed into law on July 4th will result in about 51,000 additional unnecessary American deaths each year. That bill that is now law contains one more thing that will affect, well, all of us in the future. Beginning in July 2026, there are new borrowing limits on student loans. Loans for graduate school will be capped at $20,500 per year up to a total of $100,000, and $50,000 per year up to a total of $200,000 for professional degrees. Here's the challenge. According to the Education Data Initiative, medical school students graduate with an average of more than $264,000 in educational debt, and the cost of medical school programs at private universities can exceed $300,000. So unless medical schools elect to lower their tuition, prospective students will have to turn to private loans, which typically carry stricter borrowing or repayment options. The other option is that medical school education will simply become unaffordable for some. This, of course, also carries with it an issue of equity, of who can and can't pursue a medical school education. But ultimately, it means fewer doctors at a time when there's already a shortage of physicians in this country. That shortage is even more acute when it comes to neurologists, and that impacts MS patient care. Now, to me, this doesn't sound like very good news, but I feel like I should take a moment to point out that this is not any kind of a partisan political message. These are just facts. If you see this as a good thing for families affected by MS, you should plan to vote accordingly in the 2026 midterm election. And if you see this as a bad thing for families affected by MS, well, you should also plan to vote accordingly in 2026. One more thing that could certainly have an impact on MS patient care is the 200% tariff on pharmaceuticals that President Trump said he was considering last week. Now, he also indicated that this tariff wouldn't take effect for a year or a year and a half, which is great news. And frankly, given the now you see it, now you don't White House policy on tariffs, this tariff may never become a reality. But I believe it's important for all of us to understand the policy perspectives held by the president and our legislative leaders in Congress. So this is something we'll be keeping a careful eye on. There are some encouraging results to share from a study designed to better understand the role of neural stem cells in myelin repair. But first, I want to share a little background information, beginning with what are neural stem cells? Well, these are a type of stem cell found in the nervous system, and these cells are capable of maturing into different types of neural cells. In fact, as an embryo matures, neural stem cells are responsible for forming the entire nervous system. In adults, a small number of neural stem cells persist and contribute to, well, among other things, potential repair mechanisms. Researchers have already observed that this ability to repair damage in the central nervous system lessens and eventually disappears as everyone ages. This process seems to happen more rapidly among people with MS. So, in this study, healthy mice were treated with a chemical that caused demyelinating lesions in their spinal cords. Then they received transplanted neural stem cells. The neural stem cells matured into oligodendrocytes, which is a big word to describe the cells responsible for producing myelin. These oligodendrocytes went on to directly drive new myelin formation, even in regions of the spinal cord where significant demyelination had occurred. The neural stem cells also cause cells that live in the central nervous system of the mice, called oligodendrocyte progenitor cells, to mature into myelin-producing oligodendrocytes. And the transplanted neural stem cells were observed to accelerate a response to inflammation as well. After observing this positive response to the transplanted neural stem cells, the research team transplanted human neural stem cells into the spinal cord lesions of mice that were lacking a specific protein that's necessary for the growth and function of oligodendrocytes. And the transplanted neural stem cells persisted in the spinal cords of all the mice up to 6 months following transplantation. The transplanted human neural stem cells didn't survive as well as the transplanted mouse neural stem cells, and while they still matured into oligodendrocytes and wrapped around the nerve fibers within lesions, remyelination was incomplete. Now, as I always try to remind you, there are vast differences between the central nervous system of a mouse and the central nervous system of a human. And we won't really know how human neural stem cells will react when they're transplanted into a human brain, but this is a great step forward in understanding how neural stem cells could promote remyelination. If you'd like to review the details of this study, you'll find a link in today's show notes. While we're on the subject of stem cell therapy, positive results have been reported from a study in the UK assessing the real-world efficacy of autologous hematopoietic stem cell transplantation, or aHSCT, for people with MS. In this study, the research team analyzed data from 364 people who had received a stem cell transplant in one of 14 centers across Great Britain between 2002 and 2023. 58% of these patients had been diagnosed with relapsing remitting MS, 36% had been diagnosed with either primary progressive MS or secondary progressive MS, and 6% had been diagnosed simply with MS. Median age at the time of transplant was 40, and most of the 364 people had more severe disability, with an average EDSS score of 6, indicating a significant level of ambulatory disability requiring a walking aid like a cane to walk a distance of 100 meters, or about 109 yards. In the first 100 days following aHSCT, 5 of the patients died, and these deaths were mostly attributed to the toxicity of the chemotherapy treatment that's first needed to delete the immune system prior to an individual receiving transplanted stem cells. 3 more patients died more than a year after their stem cell transplant, but these patients had significantly advanced MS, and their deaths were attributed to either disease progression, COVID-19, or other unrelated causes. Also in the first 100 days following aHSCT, 3 other patients developed new cancers, about 25 patients developed other autoimmune conditions, and about 265 patients experienced reactivation of other viruses, most particularly the Epstein-Barr virus. 5 years after their stem cell transplant, 68% of the people with relapsing remitting MS remained free from confirmed disability progression, with no progression of their EDSS scores, compared to 59% of the people with secondary progressive MS and 46% of the people with primary progressive MS. These are all pretty positive outcomes, reinforcing the belief that people who have been more recently diagnosed with relapsing remitting MS seem to be the best candidates for autologous hematopoietic stem cell transplantation. Now, if you'd like to review the details of this study, you'll find a link in today's show notes. Progression independent of relapse activity, known by the acronym PIRA, refers to the worsening of MS symptoms and disability that occurs even when someone isn't experiencing active inflammation or new lesions in the brain. And as you might imagine, because this progression occurs in the absence of active inflammation or new lesions on MRI, doctors can only acknowledge it retrospectively, after it's already occurred. So, wouldn't it be beneficial to know whether someone is at high risk of PIRA at the time they're diagnosed with MS? That's the question that a research team attempted to answer using artificial intelligence. The team developed an AI tool that would analyze an individual's initial MRI scan from the imaging that was done at the time of their diagnosis. Evaluating 259 people diagnosed with MS, 57 of whom went on to be diagnosed with at least one occurrence of progression independent of relapse activity over a little more than 4 years, the AI tool accurately predicted those occurrences of PIRA 78% of the time, or in 44 of those 57 cases. And if 78% seems a little too low to be comfortable with, please remember that clinicians currently have no way of accurately predicting PIRA. So this tool could be a very promising method of leveraging artificial intelligence to help predict future disease progression, even when that progression seems to be otherwise invisible. Now, if you'd like to learn more about this study, you'll find a link in today's show notes. I hope you've had a chance to listen to the podcast over the past couple of weeks when I featured my conversations with several of the presenters at the recent Digital Tools Workshop hosted by the International Progressive MS Alliance. If you'd like to hear more about the subjects that were covered at the workshop, the International Progressive MS Alliance is hosting an online panel discussion featuring workshop participants and presenters sharing insights from the meeting and discussing how they view the role of digital tools in the future of MS research. This live webcast will be streamed on July 23rd from the MS International Federation's Facebook page and their YouTube channel, the National MS Society's Facebook page and YouTube channel, and the MS Society in the UK's Facebook page. You'll find a link to register and links to all the Facebook pages and YouTube channels where you can watch this discussion in today's show notes. Crush MS is one of my favorite fundraisers for MS research. Besides raising money that supports the International Progressive MS Alliance, Crush MS also gives me a good reason to visit Napa Valley. And if you're looking for a good reason to visit Napa, I hope you'll join me at Crush MS on Saturday, July 26. You'll enjoy great California wines from over 24 Napa Valley wineries. There's great food, an amazing silent auction, live music, and what better way to support MS research than by spending time at a vineyard? If you'd like to learn more and/or purchase tickets to Crush MS, you can visit crushms.org, and you'll find that link in today's show notes. Research is the engine that drives progress, and we can see what progress looks like when we look at the clinical trials being conducted by Immunic Therapeutics for vidofludimus calcium. Joining me in a moment to discuss the results of their Phase 2 clinical trial for vidofludimus calcium as a therapy for progressive MS is the President and Chief Operating Officer of Immunic Therapeutics, Jason Tardio. About 8 weeks ago, biotech company Immunic Therapeutics announced the positive results of a phase 2 clinical trial evaluating vidofludimus calcium as a therapy to treat progressive MS. Joining me to talk about those results is the President and Chief Operating Officer of Immunic Therapeutics, Jason Tardio. Welcome to the podcast, Jason.
John, thank you for having me.
First, can you tell us a little about Immunic Therapeutics?
Sure. Immunic Therapeutics is a New York City-based biotech company. We also have operations in Munich, Germany, focus on developing innovative small molecule therapies to address autoimmune and inflammatory-related conditions. So we have 2 products in clinical development. We have a medicine called IMU-856, which is a modulator of a target called SIRT6 or SIRT6. We have some very interesting initial proof of concept data with that molecule in celiac disease, and we have plans to move that into a phase 2 study both in celiac and other GI-related conditions such as IBD and others. But our most advanced asset is a medicine called vidofludimus calcium, which is a very unique approach to addressing the full spectrum of multiple sclerosis. It is currently in a phase 3 program for relapsing forms of multiple sclerosis. And as you've just mentioned, we recently completed an exploratory phase 2 study called the Caliber study or Caliber trial in individuals living with progressive multiple sclerosis.
We're going to talk about that in just a minute, but I know a detail my listeners are always interested in. What type of therapy are you developing? Is it oral, injection, infusion?
Yeah, sure, John. So this is a once-a-day oral therapy. It is a unique approach to address, as I've mentioned, the full spectrum of multiple sclerosis. We know that multiple sclerosis is a disease of both neuroinflammation and a disease of neurodegeneration. So ideally, you want a medicine that's anti-inflammatory in nature, but you also need a medicine that's neuroprotective in nature to address this smoldering neurodegenerative component. There are 15 to 20 or so available therapies today that are all anti-inflammatory in nature. There's really nothing to address this underlying neurodegenerative component of the disease. And so we developed vidofludimus calcium to address both facets of MS. It's a inhibitor of an enzyme called DHODH. We know that inhibiting this enzyme suppresses the proliferation of both activated T and B lymphocytes, which play a key role in the autoimmune attack in multiple sclerosis. And we know this is a known mechanistic approach to address inflammatory disease. But it's the only medicine in development that's an activator of a target called the nuclear receptor-related 1, or NER1, which is a transcription factor predominantly expressed in neurons. And we know that this target has both neuroprotective and anti-neuroinflammatory activity. And we're very excited about the data we have because, again, we believe by addressing both NER1 activation and inhibiting DHODH, again, we can have a medicine that's both anti-inflammatory but also is neuroprotective.
I'm glad you mentioned that NER1 pathway. It is interesting. It's one that is strongly associated with Parkinson's disease and to some degree with Alzheimer's disease. How do you see that mechanism of action impacting MS?
Yes, so you're right that this target has been an identified target for a number of neurodegenerative conditions, and there's thousands of papers out there in Parkinson's and Alzheimer's and ALS. It's only the last couple of years that NER1 as a target in MS has gained significant interest. And here's what we know about NER1. We know that NER1 is downregulated in patients or individuals living with multiple sclerosis compared to healthy control. So individuals have lower levels of NER1 with multiple sclerosis. We also know amongst individuals living with multiple sclerosis, those that have even lower levels of NER1 have a more aggressive form of the disease. But probably the most interesting data point comes out of a study that was done in Italy. And as your listeners may or may not be aware, pregnancy is neuroprotective in nature. So we know that women living with multiple sclerosis who get pregnant, especially in the second and third trimester, have cessation of disease activity. So these researchers in Italy sought to understand biologically why is pregnancy neuroprotective. And they looked at individuals with multiple sclerosis compared to healthy controls, and they found that there were 347 transcripts or genes that showed altered expressions in individuals living with MS. Interesting enough, during pregnancy, 8 of these altered expressions reverted back to normal levels or healthy control levels. And one of these genes or transcripts was NER1. And so we believe, again, by activating this target, you can increase levels of NER1 in individuals living with MS and thus provide neuroprotective benefits directly by preventing neurons from cell death and indirectly by reducing activation of both astrocytes and microglial. So it's a very interesting, innovative target, and we're quite excited about what we have.
Speaking of which, what can you tell us about the results from the phase 2 clinical trial?
So we've previously reported positive results in a phase 2 study in relapsing multiple sclerosis called the EFficacy trial. From there, we moved the program into, as I've mentioned, a confirmatory phase 3 program. Those are 2 twin studies called ENSURE-1 and ENSURE-2. Those are fully enrolled, and we expect to have results from those studies in Q4 of 2026. Again, that's looking at vidofludimus calcium 30 milligrams versus placebo in relapsing forms of MS. As you've mentioned, we recently reported results from the phase 2 Caliber study. This was a study in progressive multiple sclerosis, and the goals and objectives of this study were to seek to understand whether or not vidofludimus calcium via NER1 activation would provide neuroprotective benefits in progressive patients. We hope to understand the impact of vidofludimus calcium on a variety of different outcomes, including clinical outcomes, MRI outcomes, and biomarker outcomes, with ultimately helping us to determine whether or not we want to move this program, and specifically vidofludimus calcium at 45 milligrams, into a confirmatory study in either primary progressive multiple sclerosis or in non-active, non-relapsing secondary progressive MS. So we studied nearly 475 subjects. These were individuals aged 18 to 65. They had a clinical diagnosis of either primary progressive multiple sclerosis or non-active secondary progressive multiple sclerosis, which means they had no evidence of a clinical relapse in the previous 24 months and no evidence of new inflammatory MRI activity in the previous 12 months. These patients were randomized to receive either vidofludimus calcium 45 milligrams or placebo. And as you've mentioned, we just reported the results, and they were quite outstanding. So looking at clinical outcomes, specifically at time to 24-week confirmed disability progression, what we saw was a 24% reduction for patients on vidofludimus calcium in the total cohort versus patients on placebo. Now, when we begin to separate out the different subsets of progressive patients, specifically in primary progressive multiple sclerosis, we saw nearly a 33% reduction, again, in time to 24-week confirmed disability progression for patients on vidofludimus compared to placebo. We saw a 19% reduction in those individuals with non-active secondary progressive MS compared to placebo. And we saw nearly a 34% reduction in those individuals that had active secondary progressive MS. So quite outstanding results. And the medicine continues to be very safe and very well tolerated.
I was about to say, wow. those are really wonderful results.
And so beyond that, John, you know, the data that we as a company found most compelling is the following. So collectively, in this group of patients, you're never going to have a collective group of patients that have no signs of inflammatory activity. It's almost impossible to recruit those patients. You can screen to have patients that have progressive disease, which we did. You can screen for patients that had no signs of activity, meaning no relapses, no new gadolinium-enhanced lesions, for inclusion. But then when you do that baseline MRI, you're always going to find a small percentage of patients that have inflammatory disease activity. And we did. So, for example, in the primary progressive MS subset, there was roughly 18% of patients that had inflammatory disease activity. Now, normally, in most studies that have been generated to date in progressive disease, when you remove those patients with inflammatory disease activity from the analysis, the impact on disability gets worse, right? And specifically, because this is in the public domain, I'll just comment on it. There is one product currently approved for primary progressive multiple sclerosis. That's the medicine ocrelizumab or Ocrevus. This medicine was approved for the treatment of primary progressive MS via successful results from a phase 3 study called the Oratorio study. Now, in that respective study, patients on Ocrevus showed a 25% reduction in time to 24-week confirmed disability worsening compared to placebo, and that served as the basis for the approval in this indication. But 27% of patients in the Oratorio study had presence of gadolinium-enhanced lesions at baseline, or in other words, had presence of inflammatory disease activity at baseline. And when you remove those 27% of patients from the overall results, that 25% benefit with Ocrevus actually gets reduced. And it gets reduced to basically 16%. Okay. Now, in our study, as I've mentioned, we had roughly 18% of patients with inflammatory disease activity in the primary progressive MS segment. When you remove those patients from the consideration set, we continue to see a robust 34% reduction of disability for patients on vidofludimus calcium. And so again, in the total PPMS cohort, it was roughly a 33% reduction. When you remove those patients with inflammatory disease activity, again, about 18% of patients, you still see a 34% reduction in confirmed disability protection, right? Or worsening. So, John, what does this mean? We believe that this means that vidofludimus calcium, via Nurr1 activation, is providing a real and true neuroprotective benefits. That the benefits of this medicine are going above and beyond the anti-inflammatory benefits seen with DHODH inhibition. But Nurr1 activation is driving something different here. And these data are the data that got us so excited about this program and really gives us confidence that this medicine very well may provide benefits in the future for those individuals living with both primary progressive multiple sclerosis and or patients also living with non-relapsing or non-active secondary progressive multiple sclerosis.
And what are your next steps for vidofludimus calcium?
So, John, we, given that we've just finished the phase 2, we're continuing to analyze and compile the full data set. But we will reach out to the regulatory authorities, both the FDA in the United States and the EMA in Europe, to schedule what's called an end of phase 2 meeting. In these meetings, you share your results with the regulators. You talk a little bit about your program moving forward and the desire to potentially move the program into a phase 3 confirmatory study. You talk a little bit about study design, protocols, outcomes, et cetera. And you really seek to gain alignment with both the FDA and EMA on what the next steps would be. So that's the near-term next steps, again, are to have those engagements, end of phase 2 engagements with the regulators. And then we would hope to kick off a study, most likely initially in primary progressive multiple sclerosis, probably in the first half of 2026.
That's exciting news. Well, Jason, I want to thank you for sharing such an encouraging update on Immunic's development of vidofludimus calcium, both in the relapsing remitting and progressive MS categories. We're going to look forward to hearing more news as your work continues.
Thanks for having me, John. I really appreciate it.
That's going to wrap up this episode of Real Talk MS. Real Talk MS is powered by the National MS Society. And you can share this episode of the podcast by letting your friends or family members know that all they have to do is point their web browser at realtalkms.com/411. You'll find that link in today's show notes so you can easily copy and paste it right into an email or a text. And if you have a minute, I hope you'll visit the Apple App Store or the Google Play Store and download the free Real Talk MS app for your iOS or Android smartphone or tablet. It's the best way for us to stay connected. The app will automatically download the latest episode of Real Talk MS. You'll be able to access any of our past episodes. You'll be able to save your favorite episodes. And it's a great way for me to share bonus content with you. The app is free, so I hope you'll download it today. I'm John Strum. Thanks for listening. Stay safe and make healthy choices.